Dear children, we are starting with another chapter, that is chapter 6, Principles of Inheritance and Variation. This topic has lot been discussed and still the research work is going on regarding the pattern of inheritance and how the variations are introduced. So here we are going to study about the type of variations we see and what or how the inheritance pattern is observed. So before we start with the chapter, let us first get ourselves introduced to some very basic terms. That is, first is inheritance. Now when we talk of inheritance, it is actually the transmission of traits from one generation to next. What do we mean by that? That means the traits or the characteristics are passed from the parent to the next generation, and that is what is being called it as hereditary. So we see similarity between the generations, and that is the transfer of the characteristics or the traits from one generation to another, and that is defined as heredity. The next term is variations. Variations are the differences in the characters among individuals of a species. So the differences, for example, any particular character or any particular trait is a character. So any particular trait or character, when it is observed among the members of a species, we find there are a lot of differences, and these differences only will form the variation. So variations are differences in a character, particular character among the members of a species. So that is what is variation. For example, blue-eyed, brown-eyed, black-eyed. So we have different kinds of variation in the color of the eye. That is what is variation. Genetics. It is the branch of science that deals with the study of the heredity and the variation. So genetics is a complete study of the pattern of inheritance and how these variations are introduced. Gregor Mendel, who was an Austrian, he was also a mathematician, a biologist, and he conducted series of experiments, and on the basis of his experiments he came to few conclusions. So all the research work or the basis of the genetics is based upon the work which was done by Gregor Mendel, and he was the person who first introduced mathematical calculations to explain the pattern of inheritance, and therefore he is known as the father of modern genetics or father of genetics, because he did the pioneering work. Now when we are talking of all this, how the experiments were conducted, what all things were done, that we are going to study in this introductory part of this chapter. Now, as I said, Gregor Mendel conducted a series of experiments. So the Mendel, he performed all his experiments on pea plant. That is, all his experiments were based upon the pea plant, that is Pisum sativa. Now why he chose this? It is because this plant grew very fast. Also, it was easily observable because all the characters which were shown were easily observable, and also it showed the contrasting traits. That means the traits were very clear and they were quite contrasting. Also, the plant can be self-pollinated and also cross-pollinated, and resulted into a large number of production of offspring. So the study became easy and therefore Mendel chose pea plant for all his experiments. The contrasting traits which were found by Mendel in this case was, let us first understand what were the contrasting traits regarding the stem height. He observed that very clearly the plant showed either the tallness factor, or the plants were tall, or very clearly the plants were dwarf. So the stem height was very clearly mentioned. Then the second important factor considered, he observed, was the flower position. The flower was placed either terminal, that means at the tip, or at the axillary position, that means at the side where the axil is there. Flower color. The color of the flower was very contrasting, that means either the flower were violet or white. So again, a contrasting nature of the color of the flower. Then it was the pod shape. Whether the color or the shape of the pod was inflated or it was constricted. Now these are very, very clearly demarcated or different characters which are observable. Then the color of the pod, green or yellow. Again, the seed shape, whether it was round or whether it was wrinkled. And the seed color, that is yellow or green. Now these were the characters, and these are the contrasting traits which he observed. So he chose this pea plant because of all this, and he performed series of experiments to understand the inheritance pattern. Now if we start with the crossing, or if we start with the experiments in understanding of the pattern of heredity, we start with one factor, or we are studying only one trait, that is stem height. So if we take stem height as the trait, then each of the trait is governed by a gene, and that is known as the factor. Mendel called it as a factor, but it is the trait which is being defined by a particular gene. So this was the purebred tall plant. And here, this is a purebred dwarf plant. So this is a dwarf and this is tall. Now, how do we obtain the purebred tall and purebred dwarf variety? What he did was, Mendel, he continued to self-pollinate the plants for several generations, ensuring that the next generation will be tall and the other one would be smaller. So he continued to breed and he got a purebred tall plant and a dwarf plant. Now he crossed these two plants, and as I told, these plants can undergo, the pea plant can undergo the tallness and the cross pollination and the self pollination. Now the gametes formed by these were, these are the gametes, and on crossing Now, when Mendel crossed this plants, that is purebred tall and purebred dwarf, he expected, this was cross between them, and he expected that tallness and the dwarfness will blend, and as a result the plants obtained will be all medium heighted plants, what he was expecting the plants to be in his experiments. So these are the gametes over here, and this is F1 generation or the first filial generation. I write over here filial, first filial generation. Now when he crossed these plants, he was expecting that all the plants in the first generation will be medium heighted because of the blending of the tall and the dwarf plant. But to his surprise, he found that all the plants in the F1 generation were tall. This came as quite a surprise to him because it was expected to be medium heighted plants. Now, on self-pollinating these flowers, or sorry, self-pollinating this plant, he found that the gametes form will be obviously this. Now here he selfed these plants, and on selfing we are arranging the gametes formed by the plant, by this plant into a Punnett square. This is the gamete. This is another gamete. Now, when he studied this, he found that the plants which were obtained in the next generation, that is the F2 generation, or the second filial generation, on selfing, he got a different ratio altogether. Now we have arranged the gametes formed by this in a Punnett square, and they are, when they are selfed, that means they will be forming this. Now here he found that in the F2 generation, the 3 plants were tall. So what he observed was 3 tall plants. This is, we are talking of F2 generation, and 1 dwarf plant. These are observable characters, that 3 tall plants and 1 dwarf plant. So the ratio of the F2 generation was 3:1, and since these are observable characters, morphological characters, we call them as phenotype ratio. This is a phenotype ratio for F2 generation. Also, he observed that 1 plant was dwarf. So this plant was dwarf and 3 tall plants were there. But he was not still very much sure that what is the genetic constitution of it. On further studying, he found that the tall plants were over here, 1 pure tall, 2 heterozygous tall, and 1 pure dwarf. So the 1:2:1. 1 is for pure tall allelic pair, 2 is for heterozygous allelic pair, and 1 is for pure dwarf homozygous pair. So this is the genotype ratio. This is the genotype ratio. Now, phenotype ratio and genotype ratio are different. That is 3:1 is the phenotype ratio, which explains about or which gives us an idea of the morphological observations or the outer structure which are easily observable. Whereas genotype ratio gives us an idea of the genetic constitution. As you can see over here, this is a pure tall, this is heterozygous tall, and this is homozygous dwarf. So 1:2:1 is the genotype ratio for this particular cross. It is always fixed, and here, since we are studying only one trait, we are not combining two traits, or we are not studying the two traits, only one trait, that is tallness and dwarfness, such a type of cross is known as monohybrid cross. So it is a this entire cross is a monohybrid cross. Monohybrid cross therefore can be defined as a cross which is done between the two members in the studying of one trait only, and here we are studying the trait as tallness and dwarfness. So if we try to understand and think about certain points, when the purebred tall and dwarf variety were crossed, instead of getting the medium heighted plants, the tall plants were obtained in F1 generation. So F1 generation had all the tall plants, and when we are saying this for the F1 and F2 generation, we use the term and we write filial. Filial means the offspring, that is the next generation. So here the next generation was all tall. On selfing, that means this was selfed. I write over here, on selfing or self-pollination, the following gametes were obtained, and the combination resulted into 3 tall plants and 1 dwarf plant in the F2 generation. The first generation is known as the P1 generation or the parental generation. Now here, certain points are to be remembered, that each of the trait is governed by a factor or a gene. On the basis of the study, Mendel concluded that. Also, he said that each trait is governed by a pair of genes and not by single gene. It is only after the two genes for a particular trait combine, that is they form allelic pair or allelomorphs, they will be giving rise to a particular trait. So the tallness is represented by both capital T and the other one is small t. Now when these were combined, it resulted into a combination of capital T and small t over here, as you can see. Let me use the blue pen over here. Capital T and small t all over here. But it all resulted into the plants which were all tall. So that means that there is one gene which is dominant over the other gene. And that gene which is dominant over the other gene is known as the dominant allele. Here again, we find that if the dominant gene is present, the characteristic or the allele which is governed by a recessive gene will not be able to express itself. So the one which is expressing the trait is known as the dominant gene, while the other which is hidden, which remains hidden, but it does not what do we say, it is not unable to express only, but it will express only if it comes with a recessive gene. It forms a pair with a recessive gene. So if in the presence of dominant gene, the recessive gene will not be able to express itself, whereas when it combines with the recessive gene or it forms the allele, then it will be able to express itself. And also he found that there are certain conditions which are known as homozygous and heterozygous condition, where the homozygous is one in which the allelic pair of a gene are similar, but a heterozygous is a condition in which the allelic pair of for a particular trait are different. The members are different, one capital T and small t. And here these are homozygous and this is heterozygous allelic pair. In the next video, we will be trying to understand few more terms related to the topics, and then let us go deeper into the science of genetics. Thank you.